Okay, so today we are going to talk about shigellosis. Shigellosis is caused by an organism which is called as Shigella organism. This organism is a gram-negative organism. It is non-motile, non-spore forming, and non-capsulated, which means it doesn't have a capsule which usually bacteria have outside the cell wall. There are 4 kinds of Shigella organisms, 4 species of the Shigella genus. The 4 species are Dysenteriae, Flexneri, Boydii, and Soni. These are the 4 genus of the 4 species of the Shigella genus. Now, let's see how it actually causes a problem in our body. So basically, its route of transmission is through the fecal-oral route and is transmitted by contaminated food and water or contaminated fingers. So any food or water that is contaminated with any excretory substances, it will cause shigellosis. Now when Shigella enter through your contaminated fingers, when you put those contaminated fingers in your mouth or food or water that you take in, it will go inside your body. It survives the gastric acidity, it reaches the colon, especially the distal colon, and invades the M cells and the colonic epithelial cells. After they invade through the M cells and the colonic epithelial cells, they are engulfed by the macrophages in the colon, which leads to recruitment of inflammatory cells, which release cytokines and causes acute colitis. What are the features of acute colitis? Acute colitis will mainly cause inflammation, ulceration, and microabscesses. So basically, it enters through the contaminated food, water, and fingers. It enters, goes to the distal colon, goes to the colon, enters via the M cells and colonic epithelial cells, then get engulfed by the macrophages, then is recruitment of inflammatory cells, releases cytokines, causes acute colitis, whose features are inflammation, ulceration, and microabscesses. Now, how does it basically do this? How can it invade? How is it causing this disease? It is because of certain virulence factors that it has. Now, these virulence factors, number 1 is IPA proteins, which helps its invasion through the M cells. Then there is type 3 secretion, which are needle-like projections in order to inject these virulence proteins directly into the host cell, which cause direct cell-to-cell spread and then causes actin polymerization of the host cells. There are 2 other major virulence factors that are present. One is the exotoxin and one is the endotoxin. The exotoxin that is released by Shigella is known as Shiga toxin, which is also a cytotoxin, which damages the cells. That's why it is called as a cytotoxin, which is the Shiga toxin. How does it cause cell disruption? Now, it inhibits 60S ribosomes in the cell, which indirectly inhibits protein synthesis, and it causes vascular damage of the organ. Now, this basically is how it affects the cell. This exotoxin, which is the Shiga toxin, as we saw before, there are 4 organisms. This Shiga toxin is produced by Shigella dysenteriae. Shigella dysenteriae mainly secretes the Shiga toxin. Now, let's move ahead. Endotoxin is the one which causes the inflammation, ulceration. Basically, it causes acute colitis. Now, let's see clinically. How does it affect a person clinically? Its incubation period is from day 1 to 3rd. What will it cause? It will cause watery diarrhea, it will cause fever, it will cause malaise. But it happens in 3 stages, which is first is the incubation phase, which we saw first to 3 days. Then is the initial phase where you will see all of this. You'll see watery diarrhea, you'll see fever, you'll see malaise. And then in the phase of dysentery, you will mainly see frequent small volume stools. Then you will see blood and mucus in stools. You can see severe abdominal cramps, and you can see tenesmus, which is painful straining. There are certain complications which can occur. These complications are divided into intestinal complications, metabolic complications, CNS complications, and bacteremia. Now, intestinal complications can be toxic megacolon perforations. Metabolic is hypoglycemia and hyponatremia. CNS complications are toxic encephalopathy, altered consciousness, seizures, delirium, and cerebral edema. And it also causes bacteremia. Now let's move to how will you actually diagnose this condition. Let's see its lab diagnosis. Its lab diagnosis is basically how would you transport this organism from one place to another. Suppose you want to transport this organism from the hospital to the microbiology laboratory. This organism is transported through, or the specimens are transported through sacs, buffered, glycerol, saline. Now let's see how it is cultured. It is cultured through wet mount and culture media. There are 3 kinds of culture media which are usually used. First is McConkey agar, then is DCA, and then is XLD. DCA is deoxycholate citrate and XLD is xylose lysine deoxycholate agar. Now let's see McConkey agar. If you inoculate this specimen into McConkey agar and if Shigella is present, it will produce translucent, non-lactose fermenting colonies, which you can see in this picture. If you put it on xylose lysine deoxycholate agar, you will see pink, reddish structure, red colonies with a black center. So red colonies are seen, and then if you see closely, these are the black centers which are visible. Then if you do a normal microscopic test, you'll find obviously short gram-negative bacilli which are non-motile, non-encapsulated, non-sporing. Then if you do certain biochemical tests, what are the biochemical tests that you can find out? Catalase positive, which means it breaks down hydrogen peroxide into water and oxygen. It is oxidase negative, which means there is absence of cytochrome oxidase enzyme. It is urease negative, which means it cannot hydrolyze urea into ammonia. It is indole negative. Sometimes it can be indole positive as well, which means some strains are positive and some strains are negative. Then if you see citrate negative, which means it cannot use citrate as its sole carbon source, and H2O gas is not produced. will you prevent this? So basically, if it is transporting through contaminated fingers, that is, if it is transporting through contaminated food or contaminated water, what is it that you can do? You can start with hand washing and stool decontamination. How else can you prevent this? By keeping cleanliness around you, by keeping, like, following hygienic methods around you. That is how you can prevent it. Safe drinking water, proper sewage disposal, food hygiene, and then isolation of the infected individuals is how you can prevent this. Next, there are certain other biochemical tests that you can do, such as mild of Vitek and antibiotic susceptibility testing. There is one treatment available for this, which is ciprofloxacin. So we are done with Shigellosis. Thank you.